Well, good afternoon or good evening, I suppose, to some of you. I'm Jennifer Griffin, and I'm the Director of Family Support Services at the LGS Foundation. Many of you are familiar with Tracy Dixon-Salazar, but if you're not, Tracy is our Executive Director, and also she's a neuroscientist, a geneticist, a patient advocate, and most importantly, she's mom to two adult children, Talon and Savannah, who has LGS. And on that note, I will let Tracy take it from here.
Hi, everybody. Thank you guys for joining. I'm so excited to talk about research. You know, it's one of my favorite things to talk about. So just a few housekeeping things. If you have any questions, please don't hesitate to type them into the chat box. And Jen will be keeping an eye on those. And then really we're talking about this because in about a week and a half, we're having our meeting of the minds, which is going to be a meeting of scientists for the most part, and families are going to be there to listen and learn, where we really want to challenge them to answer some of the questions that we have been asking and wondering about for a long time, those of us that have been living with LGS for a long time. So I'm hopefully going to be able to answer some of your questions about what this meeting is, what the LGS Foundation is doing in the research space to try to push research forward. Right now, the way that epilepsy, or the way that LGS is treated is that there's a focus on the seizures, but we know that LGS is more than seizures, and we want science to start looking at those questions and helping us to answer them. So this is really sort of the beginning of a big effort in this area to really start to get scientists to be thinking more about LGS and how we can design better treatments and interventions in the future that, one, don't just look at seizures only, but also are designed to look at what is unique about LGS. So I'm going to share my screen. Let me do that first. And okay, hopefully you guys can see my screen. And then hopefully when I start the presentation. All right, Jen, how does that look? Can you see my screen? Okay, I'm going to go with yes, because I can't hear you. And this is always so weird. So I'm like sitting in my office and I'm presenting to myself. I can't see anybody. I don't know if you're there, so I can't even see the chat anymore. So hopefully you guys can hear me. If not, maybe someone can text me and I'll see that. Awesome. So some of the questions that we hope to answer on this round are listed here. So I want to go over just a little bit about how we define LGS, how LGS evolves from early life seizures. Nobody's born with LGS, which I think is a common misconception. I didn't know that for many years. What causes seizures that lead to LGS, but also what causes LGS. LGS is really the abnormal brain waves that are seen on the EEG. And so what is causing that? And then I want to talk about how science might be able to help us move things along. What's happening now is more and more seizure drugs are coming out and they help a little, but they're not really helping a huge percentage of our kids. So how can we be smarter about designing treatments? And then also, what is the LGS Foundation doing in this area? So hopefully can answer some of these questions and then take a lot of questions from you guys to move us forward. So how do we define LGS? You know, it used to just be a collection of symptoms. Oh, you have a syndrome, but it's gotten a lot more technically advanced than that. This is really data from the International League Against Epilepsy, where they've broken down the way that they classify seizures and epilepsy into all of these different areas. And so I always ask families, do you know all of yours? Do you know all of your seizure types? I don't know if my pointer is visible for you guys, hopefully, but seizure types, they're up at the top in the center. Do you know if your loved one has focal seizures, generalized seizures, or is it still unknown? And then there's the epilepsy type. You can have focal epilepsy, where all of your seizures are originating in specific regions of the brain. You have generalized epilepsy, where all of your seizures are originating from the whole brain at one time. And then you can have combined generalized and focal, which is many of our LGS loved ones. And then that can also be unknown. And then maybe there's a syndrome, and a syndrome is really a collection of symptoms. And so this is the way that we did medicine for years before molecular biology and genetics and genomics and cell-based studies, is we observed children with LGS and we came to define the LGS syndrome and others. But there's also over here on the far right in red, the etiology, and that's a big fancy word for the cause. What caused the seizures to start? Everybody has seizures secondary to some cause, and it's usually a structural problem, maybe an injury at birth or something along those lines, or it could be genetic is another, and we'll talk a little bit about those. So these are, structural, genetic, infectious, metabolic, immune, or unknown. And then we also start to define the epilepsies around the comorbidities or the other associated disorders like behavioral issues, developmental delay, intellectual disability, things like that. So it's important for us as patient family members to understand seizure types, epilepsy types, whether or not there's a syndrome, but also what our cause is, and then what are the other associated conditions. And this is how we can best understand how to treat our loved one, how to make decisions based on seizure types, because this is how the doctors are doing it and how the researchers are doing it. Is they're basing it off of seizure type and syndrome type and things like that. To get a bit more specific about LGS, though, there's actually a new, more simplified. They always say it's more simplified, but it seems more complicated to me. But there's a new definition of how we're defining LGS that the International League Against Epilepsy is proposing, and it really is that tonic seizures are present and at least one other seizure type, and that the seizures are drug resistant. So that's one group of things that a person with LGS has to have. The other is an onset of seizures under the age of 18. Most kids start having seizures that then evolve into LGS under the age of 10, but it has to be under the age of 18. And the LGS itself isn't diagnosed usually until between the ages of 3 and 5. It can be diagnosed earlier. It can be diagnosed later. It can show up later. It can show up earlier. Just because you get a diagnosis doesn't mean that that's when it started. There's a lot of families who are finding out, when their kid's 12 or 13, that they've had LGS for years and they didn't know that. And that was true for us in some ways as well. So that's another criteria to get the LGS diagnosis. A third one is this very specific EEG. So the EEG is something called interictal slow spike and wave, which is less than 2.5 per second. Interictal means between seizures. So, and this is what the slow spike and wave looks like over here on the right side of the screen. This is very abnormal compared to a normal EEG, and it's present when our kids are not seizing. And then there's a new finding that's been around for about 10 years, but the definition of LGS has changed. So some of us that have, 50 and 60-year-olds with LGS probably got the diagnosis under a different definition, but it's changed a few times and they're getting more and more precise as time goes on. But now in order to get the diagnosis, you need to have this thing called GPFA, generalized paroxysmal fast activity. So this is about 10 to 20 per second of these fast activity runs. And it mainly happens during sleep. And often this GPFA is seen when tonic seizures are happening. So, LGS is really comes down to these abnormal EEG patterns, which are a sign that brain development has gone horribly wrong, that there is now brain damage. And I've never met a doctor that's like, oh, slow spike and wave, no big deal. This is a big deal. These things are bad. And these abnormal EEG features are what make the epilepsy even harder to control and what make it very, very difficult for our children to learn and to develop properly. And we'll talk a little bit more about that. And then the fourth big thing you need to have in order to get the LGS classification is developmental delay or intellectual disability. Most everyone with LGS will ultimately be intellectually disabled in their life. It can range from mild to severe. However, it's not always present at the time of diagnosis. My daughter was perfectly typically developing. She wasn't advanced. She wasn't behind when she started having seizures at 2, and she started to fall behind at the age of 3, and she didn't get the diagnosis of LGS until she was older. So you don't have to have intellectual disability at the time of diagnosis, but it is associated very strongly with LGS. So that is the definition. Now, this is the textbook definition, and our kids do not read the textbooks. And oftentimes the diagnosis is given when a child looks like they're LGS. So maybe they have slow spike and wave, but maybe it's not less than 2.5, hertz here, like 2.5 hertz. Maybe it's less than 3. right? But it's higher than 2.5. So, so there's a lot of people who get the LGS diagnosis that don't absolutely 100% fit this description. And my philosophy on that is welcome. We are your community. We are your family. We are all living the same horrible seizure journey and all the other LGS associated disorders. So you are more than welcome to be here. If you're LGS-like, or if you have a severe epileptic encephalopathy, which I'll talk about next. It is important to point out, and I just want to thank some of the doctors that have given me these slides. I've always tried to thank them in the bottom corner. This is a slide from Dr. Shepard, but there are a lot of epilepsy syndromes. So if you remember, epilepsy syndrome is a collection of symptoms, and the symptoms that are collected together in LGS are the EEG features, the tonic seizures plus at least one other type, the intellectual disability at some point, and the age of onset. But there's all these other syndromes. There's West syndrome, Ohtahara syndrome, Dravet syndrome, and you can have more than one syndrome. In fact, a huge percentage, about 30% of kids who had previous West syndrome develop LGS later. So I know that early on, it's like, well, what syndrome do I have? You can only have one. Well, no, that's not true. Ohtahara can also often evolve into LGS as well, and others. So it's important to know your syndrome for a few reasons. One is, I think it's important to find your community of support. Nobody should walk this journey alone. And it's also important because there are actually treatments that are emerging for some of these, specifically for some of these syndromes. And so you want to know what the best possible treatments are. So I'm of the mindset that my kid gets every possible diagnosis possible. You know, she's got tractable epilepsy, she has LGS, she's got calcium channel mutations that led to her seizures. You know, she's got specific seizure types, tonic-clonic, atypical absence, myoclonic. And I use all of those to try to decide, you know, on all these treatments that were offered for her. So I'm going to keep going. I'll answer questions at the end if that's the best, because I can't quite see the chat right now. So, but I do want to talk about LGS and this evolution. You know, I think that when I first heard that you weren't born with, you know, that people aren't really born with LGS, that it evolves over time. I was really shocked to learn that. And LGS does evolve. LGS is an epileptic encephalopathy, or you may have heard its phrase developmental and epileptic encephalopathy. And the short for that is DEE or EE. But in order to have an epileptic encephalopathy, you need to have seizures plus those very abnormal brain waves. And because you have frequent seizures and this frequent abnormal activity, then you get slowing of development. So the fact that our kids are having slow spike and wave between seizures, and the fact that they've got this GPA, FA, and the fact that their brains are not fully mature, that is causing them to have more problems than you would expect if they only had seizures, right? So the developmental and epileptic encephalopathy happens during the developing brain. And the best way I can explain it is that the emerging, you know, scientific research shows us that it's a wiring defect. You know, the brain is putting itself together, it's making connections in different areas. And when you have these seizures, which are abnormal bursts of electrical activity, and then you have this abnormal EEG activity, that's changing the way the brain connects itself. And then that prevents our kids from learning and developing. And that's what the emerging theories are showing us. So just to kind of drive home this idea of a developmental and epileptic encephalopathy, again, you have to have the seizures plus the abnormal EEG, and all of that together causes even more problems than if you just had the seizures and the brain was mature. You know, adults with epilepsy often don't have this evolution of seizures like our kids do, where the seizure types are changing and more than one seizure type. Adults with epilepsy will often have, you know, they'll start out with a seizure type, and that seizure type may be focal and spread, or they'll start out with generalized seizures. But there's not this evolution because their brains are mature at that point and their brains aren't developing as rapidly as these young kids are. So, you know, for those of us that have a developmental and an epileptic encephalopathy, all of those syndromes I mentioned are DEEs, LGS, Dravet syndrome, tuberous sclerosis can be, SCN8A, epilepsy, Ohtahara, West syndrome. All of us are walking a very similar journey. The science is different, but we're walking a very similar journey in terms of the doctors we see, the seizures we're dealing with, the healthcare system we're navigating, the school system we're navigating. And sometimes it's important to group us together, and then sometimes it's important to break out LGS versus Dravet versus these others. And usually it falls to the science, but not always. So this concept of, this is a very busy graph, and I'll kind of walk you through it, but there is this evolution. So over here on the left-hand side, you'll have your ideology, right? And this is time. So this is going to be a young child. Sometimes it's rare, but sometimes kids will start seizing in utero. At conception, when they were quote unquote born, they didn't have LGS, but they can start seizing in utero and then already have this slow spike and wave when they're born. But early on, what often happens is there's something that happens at birth, like a birth injury. Lack of oxygen at birth is probably the biggest cause of seizures that then evolve into LGS over time. You can also have congenital malformations. So cortical dysplasias are one, lissencephaly, double cortex syndrome. These things you can see on the MRI where the structure of the brain is somehow different than what it normally should be compared to a normal brain at that time, or a typical brain. And that suggests that something went wrong structurally where the brain was putting itself together in the womb. There are also genetic causes. So I mentioned a couple: SCN8A, SCN1A for Dravet, SCN2A. There's about 150 genes that have now been associated with LGS. So I can't list them all, but we've got kids who have Down syndrome, trisomy 21, who have seizures, who that then evolve into LGS. Tuberous sclerosis, a lot of those kids will evolve into having spasms and then have LGS. So lots of genes that are involved. Infectious diseases, so meningitis, encephalitis. There's metabolic disorders. There's autoimmune and immune disorders. And then we think that, from some of these early studies, that in about 10% of cases, we don't know the cause of the seizures. If you take a group of LGS kids and you try to figure out how many the cause in, we think we can find the cause now in about 70 to 80% of them. It's one of these things. And it's important to know your cause, your etiology, because again, treatments are coming out around these specific causes. And you want to know your options for your loved one with LGS. And so that cause will then lead to the first seizure. And you don't have epilepsy at the first seizure. Epilepsy after you've had 2 or more seizures, in its simplest definition. And up until the epilepsy starts, your brain, for the most part, it's different if you have a malformation, but for the most part, the brain has a set network. But once you develop the epilepsy and the brain is still developing, now the epilepsy itself is going to start changing the brain. And so when the epilepsy becomes treatment resistant, that's defined as having failed 2 or more treatments that were chosen and they were tried for a reasonable amount of time and they were reasonable choices, you now have treatment resistant epilepsy. Then you're even more likely now to evolve into LGS. And so you can kind of say, all right, if you have a kid who's born with an SCN2A mutation, for example, what are their chances of having seizures, right? And then if they have seizures and those seizures evolve into epilepsy, now they're becoming more likely to develop LGS. They all won't, but the odds are kind of more or higher now that they're going to develop it. And then the epilepsy, if it becomes treatment resistant, now they're even more likely to develop LGS. Not all of them will, but some of them will. And then LGS, as we've already talked about, is when you have more than one seizure type. You've got the specific EEG, slow spike and wave, and GPFA, the cognitive impairment, the age of onset, and then refractory. Interestingly, and one reason why I think it's still useful to keep, there's many reasons I think it's useful to keep the LGS terminology out there, but one is that no matter where you came from in this journey to LGS, once you have LGS, the prognosis is very similar, no matter what your cause was. Most continue to have seizures into adulthood. There's a very high risk of mortality. A lot of our kids don't make it to adulthood. Most are intellectually disabled. A few make it out. Somewhere between 5 and 10% can be intellectually normal. But we often don't see these families in our community. They often, they go on to be parts of other communities. Lots of status epilepticus, lots of injuries, lots of issues, and it's very common. And then by the time you have LGS, you've now, the brain that you started with when you first started having epilepsy has changed because the brain is developing, and now the network has not formed properly. And so, when we start to think about how science might be able to help us, it's important for us to understand what caused the seizures. But we also, there's this whole gap in the science of what causes LGS. Why do some kids evolve and to these very specific EEG patterns? Why is this pattern seen in nature, and what does it mean? And does it mean that, when you've got slow spike and wave and GPFA, that you literally are not going to be able to learn anymore? I mean, these are all questions I think that are really important to answer. So I just want to show you one piece of data here. This is a great paper from Anne Berg and her team, where they, sorry about that. Where they followed a bunch of patients for a 2-year period from the first time they were seen in the epilepsy clinic, and then they followed them over time. And they ended up following this whole group for about 10 years. But initially when they came in, 4 of them had another diagnosis, 6 were undiagnosed, 4 had LGS at the time they first visited the epilepsy center, 23 had West syndrome, 10 had generalized seizures, and 11 had focal. Then when they followed up with them about 2.5 years later, you can see how many of these red lines moved. Now 22 had LGS. And it's studies like these that show us that LGS is something that happens to children while we're watching. That's something that evolves over time. We also know that those that are most at risk of developing LGS are start having seizures less than 1 year old. And then, so about 13% developed LGS if they started seizing under the age of 1. And then some of those were infantile spasms and some of them were not. And then that percentage goes down, but it's still high if they started seizing from 1 to 2. So the younger you are when you develop seizures, there's a slight increase in the probability that you'll develop LGS. And maybe this information will be helpful to us in the future.
And then we have a question. I just wanted to
yes,
here, because I don't know if you're going to be talking about this, but this one parent asks if could you talk about puberty in LGS? And now I'm not sure what this means. Should there be a wait and see approach? Your daughter is turning 7 in January. I'm assuming it's wait and see how it evolves, or do you have any input as to, what happens when puberty hits?
Yeah, so puberty in LGS is really tough. There have not been a lot of studies on this. In fact, for a long time, there wasn't a lot of research on LGS in older children and adults because people just thought of it as a pediatric syndrome. And it's like, yes, it's a pediatric syndrome. It starts in pediatrics, but it doesn't stay there. These kids do live and they do grow up. And I do think that while I don't have really strong data on this, I think our kids are living longer. LGS kids are living longer. There are more treatment options. But puberty is a really volatile stage for a lot of our LGS families. And this would be a great opportunity for us to do some research. Often seizures will get worse. The brain is still developing up until the age of 25. And during puberty, a lot of changes are happening. Hormones have now been introduced. For my own daughter, her seizures in the very beginning, from the ages of 2 to about 7, were completely unpredictable. You couldn't predict anything. And then after about the age of, from about the 7 to about 10 or 11, they were not predictable, but certain parts of them were, right? Like I could say, okay, if she had 20 tonic-clonic seizures last night during sleep, she's going to have a bad day. Or I can say if she has a drop seizure right after waking up, it's going to be a bad day, right? So you could kind of make some guesstimates. And then puberty hit for her, and we were back to the absolute chaos. We had no idea what we were going to get. And then when puberty sort of started to taper off at about 18, 19, 20, she started to become more stable. And we do hear anecdotally of that is a pattern that's seen in LGS. What we don't know is how common that is, right? How common is it for puberty to just derail everything, all the progress that you've made on seizure control? And then what other things emerge during puberty? So, I mean, I wish I had more answers for you on what to expect. As far as a wait and see approach, if the wait and see, if you have, is for waiting and seeing if the diagnosis is really LGS, then I don't see any reason to wait. I mean, if you either have LGS or you don't, based on these criteria. And there's a lot of doctors that a child or an adult will be close. They look like LGS, and so they get the LGS diagnosis so that that child can have access to a treatment that's on the market. So that does happen too. But if it's a watch and wait, if the question relates more to, should we just watch and wait and see if seizures are going to get worse, or if other issues like behavior and things are going to get worse? I don't know of any preemptive thing you can use to treat to make puberty less intense. I do know that when puberty starts to emerge, some kids go on birth control pills, like for females, to try to regulate their cycles a little bit more. But that's usually not really done until you know there's a problem and you're trying to sort of get the hormones under control. And then I don't know of anything for males in that sense. So I wish I could be more helpful, but I'm not. So, but yeah, feel free to type more questions in the chat and I'll try to get more answers, but I'll just jump back in here. This was just the same study I was showing you earlier from Dr. Berg, where they followed these kids for a really long time. And there are 12%, 3 out of 20, there were only 24 across this multi-decade study, kids that actually ended up having LGS. It is a rare disease. But 12% of them in this study were seizure-free and off meds. We don't often see those, but in this particular study, I mean, it does happen. And this was a really nice detailed prospective study where they followed these kids for, from 1976 to 1993 and then checked back in with them. So that's what we know about LGS. What do we know about the cause of seizures in LGS? And I touched on this a little bit. I want to just show you the data one more time. Often they say that we can identify the cause in 70 to 80%. This is actually shifting now with the genomic revolution and with better imaging capabilities. In 70 to 80% of kids with LGS, we can identify the cause. And we've already talked a little bit about these causes. I showed them to you on the other slide in a different format, but so structural malformations, injury, you can also see injuries on the MRI. So they're also called structural. There's infectious diseases, there's genetics. And it's really important to know your etiology. And I can't say this enough, because there are treatments that are emerging specifically, especially in the genetics area, but not only in the genetics area, but especially in the genetics area, that would be specific to the cause of your seizures. So knowing the cause is as important as knowing the seizure type and as important as knowing the syndrome type. So I can't stress that enough. And we often talk about, my daughter has LGS secondary to calcium channel mutations. Hers is genetic. You can also have LGS secondary to lack of oxygen at birth, or hypoxic ischemia is another way that that's phrased, or secondary to meningitis. So it's really important to know that. For the structural, this is the most common. These are things you can see on the MRI where something structurally has gone wrong with brain development. Cortical malformations and dysplasias are very common, as are birth injuries. Lots of those. And then for the genetics, you can have genetic causes of your seizures that are also structural. So just because you see something on the MRI doesn't mean that it's not genetic. Cortical dysplasias are often caused by gene mutations, and you can see that also. So there was this misconception for a long time that it was either structural or genetic, but there are some that are both. There's a gene and the structural changes seen. There's also genetic metabolic. So these are things like creatinine disorders, metabolic underlying mitochondrial disorders and things like that can be genetic and metabolic. And then there's genetic de novo. So this is where the mutation arose either in the sperm or in the egg, or when the sperm and the egg came together, and mom and dad don't have it, right? And so there's been this huge explosion in the number of genes where we've identified de novo mutations, where mom and dad don't have the disease, they didn't pass it on to their kids necessarily, but their child has it. So it's really important. And if you, we're starting to build out more information on our new LGS Foundation website, where we're talking about how you can identify your cause if you need any help in this area, but this is really important to understand. So let's talk just for a few minutes about what causes LGS. And we don't really know a lot about this. And I just want to show you a little bit of the data. We know you have to have an etiology that caused seizures. So, and it could be there's hundreds of etiologies that can lead to LGS. So whatever your cause was, and you have to have these seizures, and then you have to have these abnormal EEG brain waves. And then that leads to the developmental slowing. But LGS is really a disorder of the brain network. And so if you think about the brain, kind of like the World Wide Web, right, where you've got our computer is connected to central computers. And those central compute, like we connect to the server, we connect to our Wi-Fi, and then that sends our signal, right? So LGS isn't just if my computer had LGS, my computer wouldn't just have it. It would be in that whole system. It would be in my computer talking to the Wi-Fi, and then the Wi-Fi talking to whoever it's connecting me to. And that's a network disorder. So the whole network itself is not functioning properly. So you've got areas, this area here is supposed to communicate to this area here, and that's supposed to communicate to this area here. And those three legs of communication are a network. And that whole system of communication is messed up. And there are very specific networks that are being shown to be abnormal in LGS. And there's two specifically. One is the attention network. This is the network that's on in your brain when you're actively paying attention. So if you're actively listening to me talk about seizures, then your attention network is on. There's another network called the default mode network. And this is the network that's on when you're just sitting there daydreaming. You're not actively thinking about anything. And these are not supposed to be on at the same time. You're either actively thinking about something or you're not thinking about anything and you're letting your mind wander. But there's data in LGS that these networks are on at the same time. And that is very, very abnormal. And this is really early data. It's really exciting data to suggest, because there's other networks in kids who have LGS and adults who have LGS that are not affected at all. There's other brain networks that they looked at that are fine. They look just like a typical brain, but these two networks are messed up. And if they are on at the same time, maybe it explains why our kids really struggle with learning and memory. And maybe it will give us some insights in the future about behavior and maybe about treatment. So, but what causes LGS? Why is it that some kids evolve, who have seizures and early onset and all of this develop this slow spike in wave and this generalized proximal fast activity, this GPFA? Why is it that some kids develop these things? And what do they mean? So the EEG, there's a lot of work that's being done on sleep EEGs, right? When you're sleeping, you've got REM sleep and you've got non-REM sleep. And you can take the REM and the non-REM EEG, and scientists have figured out what regions of the brain are responsible for those brain waves, and what receptors, what proteins, what molecules are responsible for those specific brain waves. And in order to understand what causes LGS, we're going to have to dig down and figure out what's causing these brainwaves and why do they develop. And if we understand why they can develop, maybe we can prevent them from developing, or we can reverse them. And I think that that's really a really important thing to think about, because we've been making medicines to treat LGS, and I'm going to talk about this in just a second, for a long time, and we've been making them the same way. We take seizures and we find something, some medicine that works, or some brain surgery or something that works towards the seizures. But when we try those in our kids, they're not having a huge effect. I mean, we tried and failed in my family. My daughter tried and failed 26 different things before we found a medicine that really helped her, that was targeted more towards the cause of her seizures. But if we could understand these brain waves and where they're coming from and why they start, maybe we can invent a smarter medicine for LGS that would eventually reverse this. We know this is bad. We know these EEG features are bad. How do we stop them from developing, or how do we reverse them? So So again, both of these networks are on at the same time in LGS, and that leads to a breakdown in the normal networks and has a significant impact on learning. And that's the biggest theory. But by and large, there's only, 1 or 2 labs in the world that are working on this question. And I think this is a really important question. So coming up for our meeting of the minds, which is taking place on September 13th and 14th for our scientists, to be a very heavy science meeting. I really encourage you to sign up and tune in, because the very first session is going to be talking, it's Monday morning, it's going to be talking about everything we know about LGS right now. And I think that's really exciting. But then the scientists are going to go into big depth detail about exactly this, like what do we need to know about LGS? When there's LGS, which is when you have the abnormal EEG features, and then there's pre-LGS, where you might develop LGS. And we want to get scientists thinking about both of those things. If you have LGS, what can we do to reverse it, stop it in its tracks, stabilize it? And if you don't have it yet, how do we keep you from getting it? So I think that, it's exciting to think about that type of a treatment as opposed to just the next seizure medicine. Now, I hope we keep getting seizure medicines, because otherwise they're just abandoning us to these horrible seizures. My kid was having hundreds and hundreds of seizures a day, and as many of your loved ones are as well. But I hope that 5 to 10 years from now, we're making smarter medicines for LGS. So let me just talk for a second a little bit about how we do treat seizures in LGS. This will probably not be news to most of you, but I just wanted to touch on this. There are 6 FDA approved medicines specifically for LGS. So they were tested in patients who have LGS. Felbamate, lamotrigine, topiramate, rufinamide, clobazam, and cannabidiol. There's another 5 drugs that they have not been FDA approved in LGS, but they've been used, they got approved for epilepsy, and they've been used and studied in patients with LGS, and they've been shown to be effective. And so your kids may be on one of these that are listed here. There's also 3 other medicines that are commonly used. They're not FDA approved for LGS specifically, but they are commonly used in LGS, but they maybe don't have the most stringent of, like, they've not been tested in an LGS population. We think they work, but we don't have data to show that. And then there's a lot of use of the ketogenic diet and other types of diets, the corpus callosotomy, brain surgery, vagus nerve stimulation, and deep brain stimulation. The thing one thing that's kind of frustrating to me is this proposed management of seizures in LGS. And we get more and more treatments, but we've been treating LGS the same way for the last 20 plus years. We throw a treatment at it and hope for the best. We don't really know which medicine works the best. We don't really know which one to use first. And so I think that these are the types of conversations that we want to be engaging doctors and researchers in the future. This first meeting of the minds is going to be really having our scientists thinking about, how do we design better treatments? How do we design something called a disease modifying treatment, something that targets the EEG or targets the prevention of the evolution into LGS? But then the next conversation we're going to have with doctors is, how do we know what to use in LGS? Does every patient really have to try all 20 plus drugs on the market and every brain surgery until they find the right thing? Or is there something we can do to understand, to learn from every patient and understand what works best for who? But the paradigm really is, you get the diagnosis of LGS. If you're a candidate for surgery, great, you can go in for resective surgery. This is a very small percentage of LGS patients. Some will be seizure-free, some will not. There's several palliative brain surgeries. Palliative means it's not going to stop the seizures, but it might help, including corpus callosotomy, vagus nerve, and now deep brain stimulation. But then most of us go this path with medication. And the medication, if you have refractory seizures, you add another medicine. And if you still have seizures, you add another medicine. If you still have seizures, you have another medicine. And if a new medicine comes out, you might be able to enroll in a clinical trial. But I hope, like I said, with the efforts that we're doing at the foundation, which we can't do without your help, and I'm going to talk about that in a minute, that 5 to 10 years from now, we're still not just throwing everything and the kitchen sink at LGS and at our kids, but that we're doing it much smarter. We've learned from the patients that have come before us, and we have a sense of which medicine might be the best in this patient with this cause, or this patient at this age, or with this patient because we see this. instead of just, oh, let's try this, let's try that, let's try this, which is very frustrating for many of us. So how can science help us? And a lot of this, the foundation has started taking a role in this, and I only have a few more slides and then I'm going to open it up to questions. But the first thing is that science needs to understand what is important to patients and families. And we've started doing some work on this, and others have as well. We hosted a patient-focused drug development meeting, which hopefully you'll be learning more about this year as we publish our Voice of the Community report, our Voices of the Patients and their families. Sorry for the cutoff there on the left-hand side of this slide, but essentially we did a survey as part of our patient-focused drug development meeting, which was really thinking about how to develop medicines and therapies for from what matters to patients. And we heard loud and clear from you that most of our families, our kids, are still having seizures. It's a lot of sleep issues, a lot of injuries from seizures. The developmental delay is a problem. Behavior is a problem. A lot of the kids have multiple physical disabilities, multiple intellectual disabilities, including not being able to read or write or even speak, and hospitalizations and the physical issues. And so from that, and really drilling down to what we heard at this meeting, where several parents testified, we learned that caregivers prioritize certain things in a certain way. We've got 100 issues we're dealing with our loved ones with LGS, but how do we prioritize them when you survey us and ask us? And seizures and safety was really high on top of the list. Developmental delay and intellectual disability, especially as it relates to communicating, our kids being able to tell us how they feel, interact with us, tell us if they're in pain, and us understanding them. Behavior was third, mobility and physical care was fourth, sleep was fifth, cost and access to care was sixth, and seven was social isolation. And it's not that all of these aren't equally important, it's just that when we said, can you rank these for us? This is what we learned from our community. And from there, we've really dug into the testimony from the patient-focused drug development and have come up with this hierarchy of needs. So any of you that ever took a psychology class in school probably remember Maslow's hierarchy of needs, which is over here on the left. And this is this idea that if you want to reach your full potential in life, if you want to be happy, if you want to reach self-actualization, that before you can do that up here at the top of the pyramid, to be all you can be, to live your best life, you need to have certain needs met. And at the basic, at the bottom, you need to have your physiological needs met, your safety needs. You need to feel loved and like you belong. You need to have self-esteem, and then you can advance to yourself, to live life to the potential, to your fullest. In LGS, we have our own hierarchy, and at the bottom is death, because most of us are just trying to keep our loved ones alive from chronic medical crises. And above that is crisis. So this would be seizures, status epilepticus, injury, infection, anything where they're closer to passing and to dying from this. Above that on our pyramid is, are we avoiding crisis? Are they basically doing the basic life things that'll keep them out of crisis? Eating, sleeping, drinking, going to the bathroom, taking their medicines, and generally being safe. And they're still going to go into crisis. I mean, my daughter would still go into non-convulsive status or get injured, even if I was doing everything right. But we spend a lot of our time just trying to keep our kids out from going into crisis. And then above that is, do they do any of those things independently? The avoiding crisis things. Do they eat? Do they sleep? Do they drink? Do they take their beds? And then most of us never make it up past that. So living their best life, it's hard. We don't even know what's at the top of this pyramid. And so this gives us a chance to let researchers know and doctors know, and also each other know, that we need to really help on this area of our kids constantly going in crisis. And it helps us to prioritize, because there's so many things that we need to do. There's so many issues that we need to address. There's so many things that scientists need to study and look at, and doctors need to help with. And so how do you sort of prioritize that? And so this gives us a chance to look at that. The next thing is to sort of understand the scientific research landscape. And we've been looking at this at the LGS Foundation, and been looking at how we've been making an impact with our scientific need. And so if you're talking to basic scientists, so these are the guys that are working in the lab, like studying fish and flies and mice and stem cells. they often think about, this is called basic science, where they're testing a hypothesis. I think that if I give this medicine to this mouse that has seizures, that this medicine will stop it, right? Or this fish that has seizures, or if I use these stem cells, then they'll tell me about the electrical activity of neurons in this patient, that kind of thing. And they're screening for treatment. So that's kind of the basic science level. And then there's the translational level, where you sort of take what you've learned from the animals and you test it in people. And that's this middle sort of translational clinical trial area here across the landscape. So something, they'll figure something out in a test tube, which is kind of the test tube here sort of represents what's the animals or something. And then you start like testing it in patients. At some point you have to move what you learned about in animals or in a study of some kind to patients, and you test to see if it's safe. And that's phase 1. And then you test phase 2 to see if it's safe and it's tolerable and if it can be effective. And then you do a phase 3, which is the double-blind placebo-controlled trial. And then that can get FDA approved. And then phase 4 is when they study it when it's out, after it's approved by the FDA and it's out in the real world. So that's sort of the clinical trial area in the research landscape. And then there's this whole area of evidence-based medicine. And this is how our doctors practice medicine. And down here under laboratory studies, this is where you would have like the animal model studies, or maybe studies of patients. Sometimes they'll study like the cerebral spinal fluid of patients, and those would be laboratory studies. And then, so down here at the bottom of the pyramid, these are the lowest levels of evidence for a doctor to treat. And as you move up the pyramid, you have higher levels of how to treat. So you can have case reports. So maybe there's something unique about a patient, somebody publishes on it, or an expert opinion. You can have case control studies. So like, hey, I'm going to try this medicine in a group of LGS kids. It's a medicine approved for, use in arrhythmias or in high blood pressure, but I'm going to try it in LGS kids. And then you can have, randomized clinical control trial. And that's this whole thing right here. You have a drug trial. So that's right up here. And then analysis of multiple trials is next, and then clinical practice guidelines. And so we've looked at where we've, looked at conversations about where are we making a difference and where are there questions across these areas. So this slide shows in the red grants that we funded in each of these areas. So we funded a lot of work to develop animal models of LGS. And we are happy to say that we believe there now is at least one mouse model of LGS now that has the EEG features, and we can kind of go in now and start to study it. And it's important. Animal research is important if you want to understand like where in the brain something is coming from and what that brain does, and you want to test things, hypothesis things. You can also study that in people to a degree. You can do brain imaging and things. You can do fMRI, you can do EEGs, you can do those things together to see where those, and we've funded some of those too over here, where we're looking at, where in the brain is LGS coming from, how does it evolve? And we've also identified where there's data. There's a lot of people making drugs for epilepsy and for LGS. So each of these areas, these in red, and then the items in clinical trials for LGS now, FDA approved for LGS now, and then some data for use in LGS. These are all active areas of research in LGS that we want to push forward and move forward. We want to understand what's being done already. But we also want to understand what the gaps are. And we now know, I have to update my slide. We now know that we have an animal model of LGS, at least one, which is very exciting. But some of the gaps that exist is that, we've talked about these, like we don't know under, evidence-based medicine, what treatment is the best. We're not learning from every patient. We don't have a treatment that targets LGS specifically. It targets one symptom of LGS and seizures. And we never even talk about all the treatments our kids are on for GI issues, for sleep issues, for behavioral issues, right? That's never even factored into the conversation with our doctor that we go see for LGS. Those are all sort of separate things. And there is a movement now to get more multidisciplinary care and start thinking about all of the other issues our kids have as a collection of LGS and not just the seizures. Seizures are a problem. They're absolutely a problem. They're a big problem, but we also have sleep issues, psychiatric issues, we've got physical disabilities, we've got the intellectual disability, and we need to start looking at those things together holistically, instead of as a piece, the way that we do now. And then also, I mean, in terms of gaps, we don't really understand how to stop all of these crises that are happening. So that's this one here, these emergencies. How can we do better? How can we keep our kids from having to go into the hospital all the time? How can we stop the nonstop seizures? What's a cluster versus what's status versus what's a seizure flurry? Like, I mean, we want to keep our kids out of crisis and not dying. So how do we address those? And these have really the gaps that we've identified are questions that families have, and they want answered. And, I think that if it's one thing for us to say, hey, we want these questions answered, it's another thing for us to go to scientists and doctors and say, hey, let's have a meeting where we actually try to answer these questions, and you tell us about your science and we tell you why this is important, and then let's all work on this together. And so that's really what we're trying to do at the LGS Foundation. And I just have 4 more slides that I'll talk about, and then I'll stop.
Yeah, we have a few questions, Tracy. So I don't know if you want to take any of those now, or you want to wait and finish your slide.
Yeah, let me just finish these last few. Sure. And I'll just go through these quickly. Okay. It's really important for us as the foundation. So the head of the NIH has basically said he would hate to have a disease where there's not a patient advocacy group. And I think that this slide is why, is it takes 17 years for something to go from scientific discovery to patients having access to that. And if we want to speed that up, I mean, you can just look at the genomic revolution. We sequenced my daughter's exome in the lab 11 years ago. The technology was identified 15 years ago. We still have families today that cannot get genetic testing for their LGS loved one. And that was 15 years ago when this technology started out in the science lab. So it's important for all of us to come together to push that forward. And we really need all of the families. There's a huge part you can play, and we're going to be talking about that. We're really the only stakeholder that's in it for the patient. We're not going to be able to find cures and disease modifying therapies without scientists and without industry partners and without the FDA and without insurance, but they all have other things pulling at them. We just care about our loved ones and are really focused on what matters to us. And there's a lot of design of things that are for us that we don't really care about. And so having a role in that is important. And then the role that we're really playing is we're working hard to update our LGS website to help you guys navigate the journey. You'll be seeing more of this as it comes up. We also have a research grants program. I hope you guys will hop onto the brand spanking new LGS Foundation website. We raised $60,000 for our at our walk this year. We've got a grant that we're going to be funding this year that we're really excited about, that we'll be telling you about soon. But we are funding research, so we're putting our money where our mouth is. We're raising money and saying, answer questions that are important to us. We do have this meeting of the minds that's coming up. It's on September 13th and 14th. They're the science days. And then September 18th is the family day. And we've got another webinar that's going to happen a couple days after the 18th family day. So you guys can ask your questions of what you learned and where do we go. And this meeting of the minds is just the beginning of several meeting of the minds conversations that we're going to be having over the next 2 years to really get our scientists, the scientists that are out there studying LGS, be thinking about the questions that matter most to us as families. And then the last thing is just this December, we have our LGS Research Roundtable where all of our grantees will be coming to talk about the research that they've done on LGS and specifically with the money that we've given them. So these are just some of the areas that the foundation is participating in to try to push the needle forward for our families. And our voice is critical. You guys are the experts. We are the experts on LGS. And we want to bring our voices to them so that as they study LGS and move things forward, they study things that matter to us. And they use our expertise to help save them time and money. And so they're not creating things that aren't going to be meaningful. So with that, I will stop and I will take questions now. Jen, how do you want to do this?
Well, I can ask you the question. Okay. The very first one is a quick one, I think. It's what is the difference between LGS and Dravet?
So Dravet syndrome, about 80% of the patients with Dravet have a mutation in the SCN1A gene. And the majority of patients with Dravet, their very first seizure is a heat sensitive seizure. And they often will have a hemiclonic, so one side of the body will seize, or a tonic-clonic as their first seizure. And they'll often have their first seizure either being placed like in a warm bath or associated with a fever, right? So often, sometimes Dravet kids will have their seizure after their first seizure after a vaccine because they get a fever, or with their first illness because they get a fever. So that's one thing. You do not see the slow spike in wave and the GPFA in Dravet kids for the most part. Now, I know numerous families who have both Dravet syndrome and LGS. But I know the scientists say that that's not possible. So I don't know what the truth is. I know that in reality and out in the real world, there are families that are walking around that have a kid that has both LGS and Dravet syndrome. But the doctors say that no, they're two very distinct syndromes. And so it's hard to say, but we all live the same journey. Like, we're seeing the same doctors, same school issues, same, medicines, all of these things. So we partner a ton with our Dravet community. What's really exciting is because there have been a number of other genes that have been identified in Dravet, but since about 80% of Dravet kids have a mutation in the SCN1A gene, there are gene therapies being developed in Dravet right now, which is really exciting for them, and for any of the gene specific epilepsies. Looking at gene therapy or targeted therapies for that genetic defect is a very exciting area of research.
Thank you. Okay, so the next question is, can you talk about some of the drugs that are currently in trial for LGS?
Yes, and I glossed over that on my slide, but I'll bring it back up really quick. There are a few on here, and we're going to be updating our website on this soon. But in trials, so Zogenix has a drug right now, it's fenfluramine, and they're looking to, it's approved right now for Dravet syndrome, and they did a trial. The trial is done, and they're going to be asking for approval from the FDA this year. So we'll see if it gets approved in LGS. There is another drug called soticlestat, and I can probably make this bigger so you guys can actually read it. It's a drug from Takeda. It had started out with a company called Ovid. And soticlestat, if I'm saying that correctly, is entering phase 3 clinical trials. And this is a really interesting medicine because it's completely different than any of the other medicines that are out there, which is really exciting. There's a trial going on for charisbamate, which, if I remember correctly, is FDA approved for other types of epilepsy, but they're looking in LGS. Eisai is doing a trial of perampanel, which is FDA approved for epilepsy, but they're looking at LGS. Marinus Pharmaceuticals is doing a trial with ganaxolone for very specific types of epilepsy. And I think CDKL5 is one, and I know a bunch of CDKL5 kids also have LGS. The RNS is in trials right now. So this is the responsive neurostimulator. You guys will be putting this up on our site in the next month or so for all of these sort of new treatments that are coming up. So you guys know about this stuff. We just got the site up, and now we're trying to get all of this really cool information in there. But this is different than deep brain stimulation. It's a responsive neurostimulator. It's implanted in the head, but it's programmed to read your seizures through EEG type technology and then to give a stimulation. Libervant is a new oral film. Some of you guys might be familiar with the clobazam oral film called Sympazam. So that's clobazam, and it's kind of like those Listerine strips is kind of the best way I can describe it. But there's moving forward to look at an oral film that is diazepam, which is a rescue medication. There's a new medicine coming out from a company called Longboard, and that's really in the very early stages. And then all of the, I can't even go into all of the gene specific therapies that are happening, like the gene therapies in Dravet or that are targeted specifically for a specific gene. But I would love to start to collect that information for our families. In the LGS population, if you have a gene, I know SCN2A is a pretty common gene, but even then, if you look in our population of about 6,000 people that are in our support group, it's really a small percentage that actually have SCN2A, right? So it doesn't apply broadly, but to whoever it does apply to, it's really important news, which is really exciting. And then this doesn't really help right now for those of us that have LGS, but there is some really exciting early treatments that are coming out, specifically for babies that have like traumatic brain injury, like lack of oxygen at birth, or specific early gene therapies. So maybe those will help in preventing, the evolution to LGS. So we are going to be putting this up on our site right now. This is our new website. And if you come to our About Research section, you can read all these really cool things. But we've got our pipeline and clinical trials page up, and we'll be adding this information up here in the coming weeks, hopefully. The motto with the new website is LGS is hard, finding information on treatments shouldn't be. So hopefully we can help you guys find quick information on what's out there. And right now we're really lucky because we do have a lot of options, and our kids need them because our kids are very, very, very sick.
And I know it's 3 o'clock, so if some people have to jump off, we are recording this and we will make it available for everybody to view. But there is just one or two quick questions. There's one about the mouse model. What is the mouse model and what is the etiology of the mouse?
So this is really interesting, and you can actually see this. If I bring this back over, you can read all about it. If you go to the For Research page, our research grant recipient, we just funded Jennifer Carney. It's a genetic model. It's KCNB1, which is a gene that can predispose to LGS. And the mouse model shows some of the EEG features of LGS. And so this model is really interesting because it could help to design therapy specifically for KCNB1 epilepsy, which I think is very important. But I think it's even more exciting for the LGS community because it allows us now to look and test theories and hypotheses in a mouse that has EEG features. So she's got one published paper on this, and we funded her this year to expand what she knows. And it's really exciting to actually have an accepted mouse model on LGS. It'll be interesting. I think this model will come up at the meeting of the minds a number of times. And so, woohoo! This is exciting. I'm excited because it's a breakthrough for us to be able to study the EEG features of LGS.
And then the last question that I have is also, it's actually about Savannah. And was there an anti-seizure drug that worked on her, or was it developed specifically?
No, it's what worked for her. So we sequenced her. We found she's got multiple calcium channel mutations, but they're in L-type calcium channels. And we know that there is a drug on the market. There was already a drug on the market that was FDA approved that was used in high blood pressure and used in arrhythmias for heart conditions. And it blocked L-type calcium channels. And so we went to our doctor and said, hey, can we try this medicine? The medicine's name is verapamil. It's been around for a long, long time, but because she had calcium channel mutations that we believed were causing her seizures, and because those mutations were letting in too much calcium into her cells, putting her on a blocker made sense, but it was a risk. And if we were wrong, we probably could have done some serious harm to her. But she was also going into status every week. She was having 300 seizures a month. She was on 7 meds, plus a device, plus a diet. I mean, she was not well. She was sleeping 16 to 18 hours a day, could barely talk, wasn't learning. So we took the risk and it worked for her. And it was a medicine that was not designed, but it was a medicine that was targeted towards the cause of her seizures. But it was one that was already on the market. And now it was never tested as an anti-seizure medicine. And I'm not quite sure why.
Awesome. Thanks, Tracy. And again, everybody, thank you for attending. I should put my camera back on. Thank you all for joining us today. And this will be available on our website, this webinar. And I look forward to seeing hopefully many of you, if not all of you, at our upcoming Meeting of the Minds and Family Day. And then we will have another look for the follow-up webinar to that if you are unable to join us. Thanks again.
Thanks everybody for joining. Take care.